You're saying that you don't have to come really close.
Yeah,
You
yeah,
have
yeah.
the distance. It
Distinguished guests, esteemed colleagues, and dear friends, on behalf of the malaria biology, parasite cell biology, and translational bioinformatics and cellular immunology teams, as well as the entire organizing committee, it's my great pleasure to give you a warm welcome to the International Workshop on New Approaches to Discover Vaccines and Drugs for Infectious Diseases here at ICGEB. This eminent event is significant because it brings together the remarkable community of scientists, researchers, and experts working on two of the most critical global health challenges, malaria and tuberculosis. It serves as a platform to exchange knowledge, foster collaborations, and inspire innovative solutions that can make an everlasting impact on these devastating diseases. This would not have been possible without the guidance and support of our PIs, Dr. Bhavan Malhotra, Dr. Asif Muhammad, Dr. Dinesh Gupta, and Dr. Dheeraj Kumar, and the Director of ICGEB, Professor Ramesh Sonti. Their strong leadership and unwavering support to research on infectious diseases continue to inspire us and drive our collective efforts. Without further delay, I'd like to invite Dr. Malhotra to officially welcome our guest and set the tone for what promises to be an engaging and insightful event. Thank you.
Thanks. At the onset, I would like to thank all the guests who have been able to make it from such long distances, and also especially our DG, who has actually been able to come. In fact, within 2 weeks, he has been able to again come and participate in the meeting. In fact, we are very grateful to him that he said, I really want to attend and participate along with the people. And along with that, I also welcome all the students who have been able to come from long distances within India as well as from various international other countries. I also welcome Sergio, who also every time enthusiastic to attend all the meetings at ICGEB and also participate in other activities of ICGEB. Thanks, Sergio, for coming to the activity. So I would like to tell a little bit about these workshops, which we organize every 2 years at ICGEB. In fact, workshops are organized throughout the year, each year, but of different areas, of the 3 different thrust areas of ICGEB. So malaria workshops are organized, I think, biannually, and this is 12th workshop which we are organizing at ICGEB, funded by ICGEB, Along with that, we have organized about 10 different other workshops with WHO, with HHMI, as well as Welcome, and so Indo-Italian workshop, Indo-French workshops. In fact, we have also organized workshop outside India under the banner Indo-French as well as Indo-Colombia. So main purpose of this workshop is actually to train the students and tell them about ICGEB activities, as well as to train about various techniques which we apply to really search for new drugs as well as vaccine against various infectious diseases as well as against other diseases, lifestyle diseases. Because as you know, there is a lot of advancement that takes place every year about various techniques. So we would like to apprise the students about some of these newer technologies which are being applied to discover new drugs as well as vaccine against various diseases that are actually coming throughout the human race development. So once again, I thank everybody, and I will invite Ramesh to tell about few words about ICGEB.
Thank you, Pawan, and good morning, everyone. Welcome to the warm auditorium and warm hospitality of ICGEB New Delhi. It may be cold and smoggy outside, but there's warmth in our hearts to welcome you. Thanks especially to Professor Lawrence Banks, our DG. He had actually said that he wants to attend this particular meeting and took time off from his busy schedule to be here. And Sergio is our friend and collaborator from the Italian embassy. As you may know, ICGEB, India and Italy are the founding members of ICGEB, and we do a lot of things together. And it is very apt, Sergio, that you could take the time. He has to go to Kerala for another meeting. An EU delegation is going to Kerala and he has to go there. So he'll be there for the first session and leave. But thank you so much, Sergio, for coming here. And most importantly, to all the faculty who have traveled from all over the world to come here. Without your presence here, your presence here really makes this event meaningful for the students. So thank you so much for those who have traveled from different parts of the world and different parts of the country, faculty to be here. And as Pawan has said, this meeting is for the students. So we really are happy that students have come from not only India, but different member countries of ICGEB. Now, ICGEB New Delhi has a long history of research on infectious diseases. We've been working on, as I was mentioned earlier, we've been working on malaria and TB for a long time, but these are not the only two infectious diseases. We also do work on dengue, chikungunya, and recently we've also, SARS-CoV-2, of course, when the pandemic was on, and recently we've started work on the Chandipura virus. So I would request the faculty who have come and the students who have come to interact with these other groups at ICGEB also to learn about the work that's going on in the areas of viral diseases here at ICGEB. Now our main focus is understanding the biology of these infectious diseases, but we also do this work with a translational component in mind. We look for vaccine candidates, and there have been candidates developed at ICGEB for developing malaria vaccines that are now undergone phase 2A clinical trials at Oxford University. Pawan Malhotra himself is working on a new vaccine candidate. We worked on dengue vaccines earlier and diagnostics for dengue and other diseases and therapeutics. Dr. Anmol and her group have developed therapeutic monoclonal antibodies against dengue, chikungunya, and SARS-CoV-2. And I know Dr. Dinkar Salunke is going to talk about his work on single-chain antibodies against malaria, TB, and also against snake bite. So there's a lot of work that is going on in ICGEB New Delhi in the area of infectious diseases. Recently, as again it was mentioned, we have also started looking at comorbidities. It's the interplay between infectious diseases and predispositions caused by comorbidities such as diabetes. So there's a lot of work going on here. It's one of the major focus areas of work in ICGEB New Delhi. And we are just happy to be able to host this workshop here. And thanks ICGEB headquarters for the support, for the Department of Biotechnology for their unwavering support to ICGEB and all our programs. And thanks again to all of you. Your being here is a source of support to us. Thank you, and I wish you all a wonderful next few days here. Thank you.
Thank you, Professor Sonti, for your warm words. Our next esteemed guest is Professor Sergio Leda. He is the Professor of Obstetrics and Gynecology at the Department of Veterinary Medicine, University of Sassari. He now serves as the Scientific Attaché, Embassy of Italy in New Delhi, India. Sir, I welcome you to say a few words to us.
Yeah, thank you very much. I think it's so nice to be here. I already expressed that when I come here, I want to stay here, to work with you, that instead of to stay in the Italian embassy. But anyway, I need to do my job. And also, it's very nice to see that you are calling me Sergio. It's not professor, it's not anything around there. That Sergio means that I'm in the family. So this is a great feeling that I have with you. Of course, I would like to thank very much Lawrence, the General Director of ICGEB, for this invitation to be here. I would like to express my thanks to Ramesh, Director of the ICGEB in Delhi, and I want to express my thanks to Malhotra Pawan because he's one of the leaders of these topics we are discussing today. So, of course, I'm not forgetting all the distinguished speakers and the people that's contributing for this meeting. I think it's already looking at the program, looks like a big success. So I just to focus on your title, it's very challenging because you said new approach to discover vaccine, drug, infectious disease. So I think your effort is to answer to two main questions. Is these two diseases still affecting a lot of people? So it's affecting the life of the people? I think the banal answer is yes. And the other question is, we have enough tools to face these two diseases, so we need to progress more. I think also the answer is yes. And probably in the workshop is quite clear that the approach should be like a multi-approach, so we cannot just looking for one phase and not the other one. This got me back in the past, because I remember when I was a child, in some walls in my country, I see this was stamped in the wall, this short acronym was DDT. So we were affected by the malaria, and they were just indicating all the places that were treated by DDT. It was a very effective approach, but was very radical, and according to my competence, was quite a lot affecting pregnancy progression and some problems. So we cannot have an effect which will be very radical. Sometimes it's very effective, but could have a big consequence. So, of course, your approach is completely different. You are trying to see also the consequence that could be related to the treatment that could be applied. So I would like to just express what Italy is doing in the malaria, because it's better, because as you mentioned, we have several collaboration between Italy and India, and of course there is like an ecosystem of institutions that are working on the malaria area. So we have mainly of this institution are related to university groups, and I would like to indicate some, the San Raffaele in Milano, the University of Padua, University of Pavia, University of Bologna, and there's some many other ones, and some other our research institutions that are face these two pathologies. And of course, each one is trying to cover a different angle of the approach for the disease. They are not cover everything, but they can put it together, they can complete the portfolio of tools that can be used to face this disease. Of course, as already Ramesh was mentioned, could be like a drug resistance to understand better the biology of the Plasmodium falciparum, or just to understand how to work on the vectors like the Anopheles and the other things. So they are, and also, of course, as according to your title, to understand, to develop new drugs, a new vaccine that could be used. On the vaccines story, I think we must say the thanks at the moment, because by WHO this has been approved the vaccine for the malaria, but probably we have some space to improve this vaccine and to have as a tool for the prevention of this disease. According to coming back also in the past, because this will be something between the modern and the past story. I remember also in my small city in Sardinia, there was an hospital that was close to the sea, and now it's interesting because now became a research institution, but before was an hospital that we called a sea hospital because we were close to the sea. And in the roof of this hospital, it was quite common, they put the passion to catch sun, and just was like a very rudimental treatment for the tuberculosis disease as well. So there were some, in the beginning, some very elementary approaches, and now it's interesting to see how much is developing the things, and now we have a new modern approaches. And just, so all these institutions are making great efforts, and also they are working in Italy, cooperating with our sanitary system, and also they are working in the European context, just because there is effort to develop some vaccine by European organization, and of course, like in international ones. Just to conclude, because I don't want to keep you very long, because I promised to speak only for 5 minutes. I don't know if I'm in time. I want to mention, because I would like to go to the past, because it's interesting to see in the past which Italians they were making some studies in malaria. And one of the scientists I want to mention is Giambattista Grassi. Giambattista Grassi was a parasitology, was working during the 80s in Italy. He was the first one who understood that the Anopheles, the vector, was transmitting the malaria. And also he tried to understand also the cycle of the Plasmodium falciparum. So he was putting exactly the base where all the people that tried to develop new discovering the malaria things. So it was an interesting story. It was quite close to get a Nobel, but you know, sometimes the scientists are fighting together, so he couldn't get the Nobel. But anyway, it's one of the pioneers studied the malaria. So from his study, a lot of the community, the scientific community, worked on the malaria and discovered all the things. So just to mention what Italy was providing from the historical point of view for the modern one. Now, coming back in the conclusion, we are here, and I think you are the best people I see the program that can develop this multi-approach that I already mentioned, and interesting to see how for different lines you are studying this two important disease. So all the contribution they are doing here would be useful for the world, and also we like to bring this contribution also to Italy, and just to say that here in Delhi is happening this important workshop that can bring scientific community to understand better these two important diseases. Thank you very much. Thank you.
Thank you, Professor Sergio, for your warm words. Our next guest is the Director General of ICGEB, Dr. Lawrence Banks. He is, as I said, the Director General of this institute, but he also heads the Tumor Virology Laboratory at ICGEB, the Triste component. Sir, I invite you to share your insights with us.
So thank you very much. Really very warm welcome to everybody to here in ICGEB. For me, it's always a joy to be here in New Delhi and to see all the friends and colleagues. And of course, I'd also really like to extend a massive thank you to the organizers of the meeting. You guys always do a fantastic job, and whenever I can make it here, I always really like to attend these conferences. I'd also really like to thank Ramesh the director of ICGEB New Delhi, who always creates such an incredible environment and a welcome for everybody at these events. And I'm also extremely grateful to Sergio Leda, the scientific attaché for Italy, who really has taken changed his schedule all around to be with us this morning. So thank you so much. So I've been tasked today to tell you a little bit about my science. I work on an infectious agent, not malaria, unfortunately, but it's human papillomaviruses. And what I'm going to try and do when I give that presentation is to try and give you a little bit of a feel for some of the approaches that we're taking to try and develop novel therapeutics, and which I think will hopefully be relevant for you that are probably interested in other infectious diseases. But before I do that, I was also asked to say a few words about ICGEB. I know for many of you, you probably know quite a lot about us, but for those of you who don't, I think it's worth just highlighting a few points about the organization that may be of interest to you and which you can probably maybe take benefit of when you go back to your home labs. So we're an international organization. We have currently 69 member countries located throughout the world, and one of the things that's important to remember is that we're actually located in 3 centers. We've heard about Trieste in Italy, and obviously here in New Delhi, but we also have laboratories based in Cape Town in South Africa. And the mandate of ICGEB is very simple. It's to be a center of excellence for research and training in molecular biology and biotechnology. And the key point there is addressing the needs of our member countries. And so really, this is something I really want to hear from you, especially for those of you who are from outside India. Where can ICGEB help you? That's what we're here for. And everything that we do is obviously linked to the SDGs in one form or another. And what I'll do in the just next few slides, I will highlight where we cross-cut on meeting the Sustainable Development Goals. Now, one of the things that's unique about ICGEB, there's many agencies similar to us who work in the development sphere. But we're unusual in that we actually have research labs. And so that's absolutely something that makes us unique. And it means we have the scientific expertise to address many of the problems that the world is facing. But we don't do it alone. We do it in collaboration. And we work very, very closely with scientists from our member countries to do research that's relevant for many of the global problems. I should say that within ICGEB as a whole, we have scientists from around 50 countries from throughout the world. So it's really quite impressive. Just to quickly say a little bit about the activities. Obviously, infectious diseases, and you're going to hear a lot about that over the course of the next few days. But we also have a large group of scientists working on non-communicable diseases, ranging from cancer to neurodegeneration, cardiovascular disorders, and the development of novel gene therapies. So we have really quite a broad remit in the health sector. Then we have scientists working in medical biotechnology, and I'll dedicate a little bit of extra time to that towards the end of this presentation. Here in New Delhi, we have a fantastic group working on industrial biotechnology and the development of second, third generation biofuels, as well as carbon capture technologies. And again, I'll say a little bit about that towards the end of my presentation. And then we mustn't forget, we're all working to make healthy populations within our communities. But if you have healthy populations, you need to be able to feed them. And so we have a very active programs in the agricultural sector dealing with studies looking at crop improvement, whether it's resistance to various forms of biotic or abiotic stress, development of novel biofertilizers, as well as development of biopesticides. So really, ICGEB covers really quite a broad area of scientific endeavour. From that, then we can lead into the various programs that we have, which is based on that science. Obviously, education is a very important one. It links to SDG 4, and this is sort of attained through various varieties of PhD and postdoctoral fellowship programs. And something I'd like to stress, we're incredibly committed to gender equality. This is incredibly important. In many of the parts of the world where we work, inequality and gender balance is something that we really work hard to try and address. Just to give you a feel for the scope, in 2024 we've had over 400 people on board in various programs. Many of those are on external support, but it's really a very vibrant fellowship program, and as you can see, we have people coming from all over the world to work in our laboratories. We also support a program of active South-South cooperation. So any of you, if you have research projects where you identify a lab in another ICGEB member country where you feel that collaboration will be important and informative for your research, talk to ICGEB. We can support these types of fellowship exchanges. One of the educational activities that we take part in is obviously the organisation of meetings, courses and workshops, just like this one. But we routinely have around 30 such courses that we run throughout the world each year. So again, if any of you or your PIs are interested in establishing a workshop, please do get in touch with us. Finally, we are also a grant funding agency. We provide direct research support to laboratories in member countries. And this is a pretty big program. This is just to show you the number of projects that are currently ongoing. 102 projects in 42 countries. It's a big investment from ICGEB, but it has a massive global impact. Obviously, one of the things that's incredibly important, we don't want the science to be done in an ivory tower. I'm a great, passionate believer in blue sky science, but we also want science to get to the people. And so this is SDG 9. It's getting the fruits of modern molecular biology and biotechnology actually to the people who need it. One of the programs that we have is a biodevelopment unit, which is based in Trieste, which develops products for direct transfer to industry. These are just some of them, but for example, these are products that are produced to a level which meets the standards of the FDA and European Pharmacopoeia, for instance, for treatment of diabetes. And those technologies are transferred directly to industry at an incredibly reduced cost. And we have over 118 such transfers have taken place. And what it means is that you can actually get the products to market so that people can afford them in low resource environments. And I think that is incredibly important. This group is also very active in training people in biopharma production processes. So if any of you are interested in scale up and how to determine whether the product that you're developing is of potential biotherapeutic relevance and can actually be scaled up for pharma production, you need to speak to us about the processes for doing that. Moving on, I'd just like to highlight something that I think is also incredibly important when we think about tech transfer, and that is developing processes for industrial development. In this case, looking at energy and production of environmentally friendly biofuels. And this is work that's been done here in New Delhi, which I think is incredibly exciting and is really going to make a big impact in terms of making the processes of biofuel development affordable in many of our member countries. Not forgetting some of the activities in Cape Town. This is a program developing biopesticides and ensuring reduction of pesticide residue in late season crops. That's incredibly important for countries that want to market their produce in Europe or North America, you have to reduce those pesticide levels. And so this is a big program within the whole of Southern Africa to help them in meeting those challenges. And something that might seem really, to somewhere like India, it's not really relevant, but for many parts of the world, access to reagents for doing molecular biology is a real challenge. I travel a lot, and one of the most common things I hear is people say, oh, we'd love to do these experiments, but we have to wait 6 months before we can get the reagents. I mean, this is a killer. It's a disaster. And so one of the things that we're doing in Trieste is developing toolboxes so that people can produce their own molecular biology reagents. And this really does shortcut that whole, the delays that people can encounter in doing research and building up their own local capacity. So with that, that's my institutional hat. If anybody, what I like to think, what we do as an organization is really to do science to promote global development. Does anyone have any comments, questions, or shall I move on to the science? Okay? Okay, let's say a bit about the science. How are we doing for time? Okay, so what I'm going to do now is tell you a little bit about some of the work that we've been doing on human papillomaviruses, major cause of many human cancers, and my interest is really identifying novel targets that we can use for therapeutic intervention. One important point to note is that many human cancers are caused by infections, and around 12% of all malignancies have a viral etiology. So that's really very important, especially when we consider that in many cases you can develop vaccines against viruses. So, vaccination against viruses can really have a major impact on cancer prevalence. And this is just to show you the 7 different virus families that are associated with cancer development in humans. I'm not going to go through the list other than to just say that for many of these viruses we have vaccines, such as, for instance, hepatitis B. That is having a massive impact on liver cancer development in many parts of the world because of the vaccination. And the same is obviously true also for human papillomaviruses. 2 other points to take from this slide. There's not one specific type of virus which causes cancer. As you can see, they come from all different families, double-stranded DNA, different forms of RNA viruses. They cover a whole different span. So there's not one particular single characteristic that you can say, oh, that's why this virus causes cancer. There is a commonality, though, amongst them. All of these viruses, once you're infected with them, you have them for the rest of your life. So that is the key point. These are viruses that are often quite slow-growing. They remain persistent. And persistence is the key word in viruses and cancer. It's because they induce a state within the infected cell, whether it's through chronic inflammation or whether it's through induction of DNA damage, that they provide a driving force for the eventual development of a tumor. And what I'm going to do is I'm going to use human papillomavirus as the example to show you how some of those features actually result in the development of malignancy. Now, human papillomavirus is arguably the most important cause of cancers caused by an infectious agent. They account for around 100% of all cervical cancers. A very large proportion of other anogenital cancers are caused by these viruses. And there's a really massive expansion in the numbers of head and neck cancers that are being caused by these viruses. In fact, if you think about North America, head and neck cancer caused by human papillomavirus is now an even greater problem than cervical cancer. So we're talking around 700,000 new cases each year. So it really is a massive global problem. And what's important to note, however, is it's only a small subset of this virus family that causes cancer. And they're listed here. There's 12 different virus types, the most important of which are these two guys, HPV-16 and HPV-18. They account for around 80% of all cervical cancers. And in actual fact, this virus, HPV-16, is actually the most oncogenic biological agent on the planet, which is really quite remarkable if you think one virus is such a potent carcinogen. And so what I'll be doing is I'll be focusing my attention primarily on those two virus types. Now, why would an organization like ICGEB be interested in HPV and cervical cancer? Well, if you look at the global distribution, you can see that it really is, this is the distribution of cervical cancer overlaid on the ICGEB member countries. And you can see it's really a desperate problem in the global south. Within Southeast Asia, HPV and cervical cancer is still almost at epidemic levels. And within Africa, the numbers might not look so great, but it's actually even worse there because we have chronic levels of underreporting. We really don't know how many people are dying from cervical cancer in many parts of the African continent. This is a complex slide, but it's really just to emphasize that human papillomaviruses, it's a huge family. There's over 200 types that infect humans, and I just told you there's only 12 that actually causes cancer. Every single person in this room is infected with human papillomaviruses. We all have multiple different types on our skin, and we never know we've got an infection. Because most of the time, these viruses are in complete balance with the host, and you never know they're there. They just go about their business, they slowly replicate, they shed, they'll infect somebody else. No one ever knows, it's not a problem. But for some reason, there's a group of viruses that we call high risk, which consist of those 12 types that I told you that actually cause cancer. And that's what my lab's interested in. We want to know, why do these virus types cause cancer, whereas all the other types do not? And to begin to get an understanding of that, we have to look at the viral life cycle. So this is the skin. This is an epithelium. It can be cutaneous or mucosal. And the skin is made up of multiple layers of different cells, okay? You have a single basal cell, which is located here. This is the only cell in your skin that can divide. And when that cell divides, it pushes its daughter cell upwards. And so it's a continual process that makes up the multiple layers of the skin. And it's this multiple layer, this so-called differentiation, which is absolutely critical to infection for human papillomaviruses. Most viruses go into a single cell, replicate like crazy, and then lyse the cell and spread virus to everybody else. Human papillomaviruses, they get access to these basal cells through small lesions in the skin. And then as you get this differentiation process, you see coordinate expression of the different viral gene products, which ultimately give rise to new infectious virus particles. So the key point here is, we're not going into one single cell and producing a lot of virus. We're going to a cell which then changes as you go through the epithelium. And so the environment in which the virus replicates is continually changing. So it's quite a challenging environment for the virus. The other thing that's critical to highlight is that the virus amplifies its genome here in these mid-epithelial layers. In a differentiating epithelium, these cells have no machinery for replicating DNA. They're in a G0 type state. And so what these two proteins, E6 and E7, do is actually they push these cells back into a kind of pseudo S phase so you can replicate the viral genome. And so this is what it looks like. This is normal infection. You produce infectious virus. It takes the time for the life cycle, takes the same time as it takes for these cells to migrate up through the skin, which is around 2 to 3 weeks. Normally, these infections are cleared after a couple of months by the immune system, and that's it. And many people don't even know they've had the infection. But in certain cases, you get persistent infection, which can last for several years, where the virus is continuing to grow, it's continuing to be expressing these different viral proteins. And this is the single biggest risk factor for ultimate progression to malignancy. And what happens is that over time, you lose the ability to replicate. All the cells enter a process of division and multiplication. And this whole process is being driven by these two viral proteins, E6 and E7. So it's like a kind of mistake. They want to drive replication and DNA replication to replicate the virus, but if that process breaks down, these two viral proteins can ultimately contribute to driving malignancy. And this is a mistake. This is not what the virus normally wants to do. What do these lesions look like? This is a cervix. And on the cervix, you can see here there are two lesions. And first of all, what I'd like to do is focus on this one, which is barely visible, very small acetowhite staining. And what has been done, we've taken a section through this lesion, and this is the section, and we stained it in two colors. One in red to show cells that are replicating DNA, and the other in green to show cells that are producing late virus products, new virus. If you look in the area that's uninfected, the only cells that replicate DNA is this basal cell, which is exactly what you'd expect. But when you look in the region which has HPV, look at the numbers of cells that have all gone back into an S phase and are replicating DNA. Cellular DNA is being replicated, but from the viral point of view, what's most important is the viral DNA that's being replicated. And this is all being driven by those two viral oncoproteins, E6 and E7. This is the late gene products being expressed, producing new infectious virus particles. So this is a normal infection. This is what HPV-16 wants to do, the most oncogenic biological agent there is. But this is how it normally grows. If you then go on the same cervix, you see there's another much more prominent lesion here. You do the same analysis, and what you see here is that where you have the infection, there's a massive expansion of the numbers of cells that are replicating DNA. And within here, you can see aberrant mitoses. There's all sorts of weird things going on. It's basically a pre-malignant lesion. And you can see the virus is actually losing the ability to replicate itself. It cannot produce late gene products because you're losing that ability to differentiate. And this is a high-grade cervical lesion. If this was not treated, it would develop into full-blown cervical cancer. And so this is the model of how we think these viruses cause cancer. You have an initial infection, you produce new infectious virus particles, the infection is resolved, and there's most likely a latent type state which this virus takes up. But in certain individuals, you have a long-term persistent infection which can allow genetic changes to take place in these infected cells, which can ultimately allow progression to, they offer proliferative advantage and ultimate progression to a high-grade lesion, just like the one I just showed you, which can then ultimately progress to malignancy. And so my lab's been interested in, for many years, how, can we look at how these two viral proteins, E6 and E7, work? And by doing that, can we identify novel ways in which we might be able to intervene? We know that we absolutely have to have these two proteins expressed in the cancer for the cancer to exist, okay? For instance, you can take HeLa cells. HeLa cells have been grown in the lab for over 50 years. They were isolated from a woman with cervical cancer back in the late 50s now. And they have HPV-18 DNA present. They express these two proteins, but despite 50 years grown in tissue culture where the genetics of these cells has gone crazy, you can still block expression of E6 and E7, and those cells will cease to proliferate and they'll die. So really, these two proteins are excellent targets for therapeutic intervention. And what I'm going to do in the next sort of 10 minutes or so is say a little bit about the sorts of approaches that we're taking with these two proteins to try and identify relevant targets and how we can potentially intervene. So this is E6. I've spent longer working on this protein than anything else in the course of the last 30 years. And it's like all of these small DNA tumor viral proteins. They're incredibly multifunctional. And that multifunctionality is linked, A, to the fact that the environment that they're in is continually changing, if you think about that differentiating epithelium. So they have to do lots of different things as you go through that environment. But also, they have no enzymes of their own, this virus. It's got to interact with many different cellular targets to basically make the cell do what the virus wants. And many of these targets, and it doesn't matter what they are, most of them are really, the associations are to help the virus replicate. That's what the virus wants to do. And so you have activities related to genome amplification, genome maintenance, you have activities related to apoptosis inhibition, which is all things that the virus needs to deal with in its replication. The byproduct of some of these interactions are things such as linked to cancer development, telomerase activation, and basically the capability to make the cell immortal. Now, one of the things that we've been interested in is to try and understand why do some viruses cause cancer and some viruses do not. And if you look at the E6 protein, this is the very C terminus of E6, and these are all viruses which cause cancer. You can see there's actually a very highly conserved stretch of 4 amino acids at this C-terminal region of the E6 protein. And this is what's so-called a PDZ binding domain. It interacts with cellular proteins that contain PDZ domains, which are like it's an 80 amino acid pocket. The C-terminus of that slots into that pocket. And basically, many of these proteins are involved in the regulation of processes such as control of cell-cell attachment and control of cell proliferation. Low-risk virus types, such as these shown here, completely lack that sequence. So this is almost like a molecular signature of oncogenic potential. And what we know is that this sequence is diverse amongst those viruses. You can see it's not identical. There's a lot of similarity, but it's not exactly the same amongst them. And when you look at it in detail, you realize that even very minor changes in this extreme C terminus can have a profound effect upon how this viral protein works. And I'm just going to show you an example of two different substrate proteins. It doesn't matter what they're called. They're both involved in the control of cell polarity. So that's how a cell knows the top from its bottom when it's in that process of differentiation. And you look at this protein, this is E6, 16E6. It can degrade this protein, as you can see here. But it doesn't do it as well as 18E6, which does it much better. And that's all due to the change in this last amino acid. You swap them around, and actually you can make 16 work a lot better, and you can make 18 less efficient. And it's actually the other way around with this other substrate. So what I'm trying to say is that very minor changes within this target sequence by which E6 selects these substrates can profoundly affect how it can target cellular proteins that it's associated with. So what we did was to extend this analysis using a proteomic screen where we asked a very simple question. These viruses are all group 1 cancer-causing viruses. They all have this very highly conserved C-terminus. We have another group here, very rarely found in cancer, but it does happen. They also have this conserved sequence, but it's beginning to change quite a lot from these guys. And then we have this virus, HPV40. This is benign. It never causes cancer. The sequence here is very, very different, but it nonetheless does have certain characteristics that fall into that, that will classify it as a PDZ domain. We did the proteomics, and I'll just show the results from what I think are the most critical virus types. These are all the cellular proteins that we identified interacting with this sequence. And what you can see is that with HPV16, it's incredibly, it's got an incredible ability to interact with multiple cellular targets. So it potentially can perturb multiple signaling pathways. The same is also true with HPV18. But if we go to HPV66, which you find extremely rarely in cancers, the numbers of targets is greatly reduced. And then if we go to HPV40, which we never find in cancer, it essentially only targets one cellular protein, this one here. So what we think is happening here is this is the precursor virus. This is the older virus. This is the one that evolutionarily came first. And so it developed this motif, presumably to allow interaction with certain subset of cellular proteins. But over time, this motif has evolved such that these virus types have really gained a much broader activity and can target many more different cellular pathways. And so this is really just to summarize that. This functional flexibility within this motif seems to be very important with regards to the ability of these viruses to contribute to cancer. And obviously one of the things that we're really interested in now is using this very highly defined interaction of which we now have structural information to develop novel ways of blocking the association between E6 and these classes of cellular targets. I'm going to finish now with just a few words about some of the work that we've been doing on the E7 oncoprotein. Again, incredibly multifunctional, interacts with a whole range of different cellular targets. Again, it doesn't matter what they are. Just to stress that the main activity of E7 is to push the cells to replicate DNA. That's what E7 does. It essentially drives cellular proliferation. And what we've been interested in is these two amino acids here, which are basically a phosphoacceptor site. And the reason we're interested in this is that there's two approaches to try and inhibit these viral functions. The one I just showed you on E6, we're thinking about small molecules that can block a protein-protein interaction. In this case, what we're thinking about is, well, can we target the cellular enzyme instead? The cellular enzyme which actually phosphorylates the E7 protein. And so that's the two-pronged approach that we're taking. And so what we've been interested in is this motif here. This is the region of E7 with the two serines here, and it's just downstream of a stretch of amino acids here, which confers interactions with proteins that drive cell proliferation. And what we've been interested in is to know whether or not these serines here are important in cervical cancer. We know from many studies in vitro that we can mutate this region of E7 and it cannot cause cell transformation or malignancy. But what if we take cells from a cervical cancer? Are these two serines still important? And so what we've been doing is to do genome editing technologies such that we can block the phosphorylation of E7. And that's what you see here. If we mutate these two serines, you lose the ability of E7 to be phosphorylated. And so we've introduced those mutations into cells derived from a cervical cancer. And when we did this, and we looked at the proliferation of those cells, we were initially quite depressed. This is under high serum. So this is a rich nutrient environment. The mutant cells maybe don't reach quite the same level of saturation density, but they grow pretty well. And we thought, oh, okay, the virus, this is not going to be a relevant target. But then when you go into low nutrient conditions, where we put the cells in low serum, the wild-type cells still grow just as well. They're fine. The mutant clones grow much more weakly. And this is really important because in a tumor, it's often in a really nutrient-starved state. So this is an aspect that we feel is really relevant for potentially treating a tumor, in that the E7 protein is creating an environment that can allow cell proliferation under low nutrient conditions, and that's really key. So how is it doing it? What's the molecular basis? I mentioned originally we have this interaction site here, which is a protein that drives cell proliferation. But I had a brilliant postdoc in the lab from Nepal who was looking at this sequence, and he identified here another overlapping motif, this tyrosine, any amino acid, and a hydrophobic, okay? Just upstream from this phosphoacceptor site. And what he realized was this motif is actually a recognition site for a different class of cellular protein, proteins called AP2, which are all involved in regulating endocytic transport, something that we really didn't expect. And when we look back in the literature to an old paper from some of our colleagues back in 2012, we realized that these AP2 proteins were actually interacting, or potentially interacting, with the E7 oncoprotein. And we could actually confirm that in a simple pull-down experiment. The interactions are direct. That's very important. Use purified proteins to show that the association is direct. It's not an indirect part of a complex. And we also know that you absolutely need an intact phosphoacceptor site for that association to take place. So we've got a component of the endocytic transport machinery interacting with E7 that requires phosphorylation. And this is the part of the cell that we're talking about here, clathrin-mediated endocytosis. I told you that it's an overlapping interaction motif, in that we've got RB binding, we've got the AP2 binding site, and both of which are regulated by casein kinase 2 phosphorylation. How do we separate them out? Well, we did a lot of mutational analysis, and just to highlight two key mutants. There's this cysteine to glycine mutation here, which is this cysteine here. We make this mutation, and we retain interaction with AP2. However, that mutation kills interaction with RB. Okay. The tyrosine here, just one amino acid further down, we make that mutation, you kill interaction with the AP2, but you retain interaction with the PRB. So we've been able to dissect these two interaction modules within the E7 oncoprotein. How does this affect the biology, and how does this correlate back to regulation of cell proliferation? We looked at cell surface expressed proteins where E7 is being expressed, and we were looking for changes in proteins that would be maybe involved in regulating cell proliferation on the cell surface. One of the proteins we identified was the epidermal growth factor receptor, which is a key regulator of cell proliferation in epithelial cells. And what we found was that EGF receptor signaling in control cells, when cells get stimulated to grow, EGFR gets hyperphosphorylated and you have a signal for a few minutes and then it gets turned off because the cell doesn't want to overproliferate. When you have E7 there, that proliferation is maintained because the signaling of the EGF receptor is continuing. With the mutant that can no longer interact with the AP2, the effects on EGFR are very much like the control cells. So what we think is happening is that this ability to interact with the endocytic transport machinery is driving EGFR receptor signaling. And that is indeed what the case is. It's actually what is happening is E7 is inhibiting the association between endocytosis and the EGFR. So the EGFR stays on the cell surface and it drives proliferation. What's the biology of that? Well, basically, if you go into cells under low nutrient conditions and you do these cell transformation assays, you find that the ability to associate with the endocytic machinery is absolutely essential for the ability of these viruses to cause cancer. So what does this mean and where are we going? For me as a virologist, point 4 is the thing that I think is really amazing. We've taken a very small stretch of amino acids within this viral protein with what we thought had only one interacting motif, but actually it's got two motifs that are overlapping. One of which confers interaction with the class of proteins in the nucleus, which also contributes to inducing cell cycle progression. But we also have another class of targets when the viral protein is in the cytoplasm, which is driving cell proliferation through receptor, through growth factor receptor signaling. And of course, moving forward, there are now clinically validated casein kinase 2 inhibitors, which we're using in cells derived from cervical cancers to see if we can develop novel forms of therapeutic intervention for treating HPV-induced malignancy based upon these activities of the E7 oncoprotein. This is the lab in Trieste, and it's an old slide now, but Om Basakula was the postdoc that did all the work that I was telling you about just now about the E7 oncoprotein. This is the current lab, and one of the things I'd really just like to say it's fantastic about being in ICGEB. You can see really it's like it reads like the United Nations. We have people from all over the world, which is really great. And again, this is what we're about. And that's my email if anybody ever has any questions. So thank you so much.
Hello.
Hi.
Is there any crystal structure available for the E7 protein from which we can get some idea that how changing the terminal amino acid from 1 to causing cancer or no cancer?
Yeah, okay. So unfortunately, within that particular region of E7, what we're trying to do is to get that region of E7 is highly unstructured.
Right.
So it's intrinsically disordered. So we cannot we cannot get the structure of E7 in that part by itself. What we have to do is to do co-crystals. And so there's already a structure available for the E7 with the retinoblastoma. And what we're doing now in the lab is to get the structure of it with the AP2 subunit. And when we have those two, we should be able to answer that question.
Okay. And how does this virus causing the persistent infection? And there is any treatment for that infection?
Okay, the persistence is essentially due to immune evasion, okay? And there is no treatment. That's it. You know, there's a vaccine which will prevent infection, which is really, really good. But once somebody's infected, it's not a therapeutic vaccine. It won't clear the infection. And so the treatment's really, it's basically surgical ablation to remove the lesion is really the only treatment that's available. And obviously, when you get to late stage cancer, it's chemotherapy and radiation, but with not great success.
Thank you.
Lawrence, have you really identified the whole complex which E7 makes within the cell? Because see, there are two independent functions, but they are very close sites.
Yeah.
So one is like even RB is known to be tumor stressor sometimes, but it is taking it to nucleus for application, and other is taking into the outside. So there are two independent functions.
Absolutely.
Is there any competition between these two sides? And maybe at one time one sets up is binding is happening, another time is different binding.
Okay, we know that both interactions take place around the same time, in that there's that CK2 phosphoacceptor site, which regulates both, is most heavily phosphorylated as you go through G1 and into S phase. We also know that we have the two pools of E7. We have a nuclear pool and we have a cytoplasmic pool. We don't know really how that shuttles, so that's something it's something we don't know. But it's clearly two separate locations of the protein.
So my question is whether they are independently, they are at different times these complexes are forming, are doing different
I mean,
roles
they can't form simultaneously. Now, you're going to have one pool of E7 in the cytoplasm, which is associating with the AP2. If you try to mix them in vitro and make you can't. I mean, they'll inhibit each other. We haven't done the direct comparison of relative strengths of association, but they're so overlapping, they're not compatible with a triple complex.
Very nice talk. Can I go? Yeah, so very nice talk. I was just trying to visualize the cell cycle machinery operating in tandem with E7. In that context, how does the phosphorylation of retinoblastoma protein, which is critical by CDK4, CDK6 for G1 to S transition, how does it go hand in hand with E7 association?
Yeah, okay. So basically what is happening is the RB should be phosphorylated and is interacting with E2F and blocking cell cycle. Right. What E7 does, which I didn't have time to say, is actually E7 specifically interacts with that phospho form of RB and targets it for proteasome degradation. So the RB gets degraded and then releases the E2F.
In that context, quick question. I mean, does this virus infect terminally differentiated cells, such as neurons, which do not have any cell cycle activity? And could that be the reason? No.
I mean, it's... Actually, the virus can get into a lot of cells, but the restriction point for it is its promoter. So you can infect fibroblasts, you can do all sorts of things with it. The virus will go in, but you won't get any gene expression because the promoter is so epithelial specific.
Lawrence, very nice talk. I also had the same kind of curiosity that this girl asked you, and you made me more interested when you said that region is flexible, that motif where one amino acid change, which is also from valine to leucine, which is both hydrophobic.
Yeah.
And it would be nice actually if you have a structure in complex with the receptor domain. But in the meanwhile, it would be interesting to see now alpha fold region, if that structure should
even if you put it through alpha fold, it comes out as disordered. you know, there's really, yeah. You know, you can try modeling it. We've done it. We do have AlphaFold where we've put it in context with the AP2, and you do get something. But I want to see the real thing.
There is a huge substrate specific difference, and it decides whether it is carcinogenic or not. So that is a big change.
Yeah, absolutely. Sergio, there's another one down here.
Yeah. I've got the microscope. I've got the microphone meanwhile, so maybe I can start. I'm right here.
Oh, sorry.
So it seemed that just one mutation would change the interaction with other cellular partners. My question is, are there any correlation between mutations such as that and infections that don't result in cancer?
Okay, so the virus itself is incredibly stable. So HPV16, for example, that sequence I showed you is the only time, that's the only sequence we ever find. It's not like some of the RNA viruses which are changing all. The papillomaviruses, you get very low frequency of mutation. And within these critical domains, we've never seen a mutation. So I don't think it doesn't seem to happen. So all you can do is you can look at the different virus types that have evolved over millennia and comparison, but there's other things as well.
I guess also there isn't necessarily screening when people don't get cancer. But then the other question is that a potential gene therapy approach?
Okay, so there's actually a huge amount of sequence data on, especially with NGS now, and where you can actually look in people that don't get cancer. And although you can see changes, occasional mutations, but it's mutations associated when you've got high-level DNA replication taking place. In terms of, and they're thought to become abort infections. In terms of what becomes a transmissible element at the end of it all, they all have that sort of basic sequence. There's no mutation. In terms of gene therapies, in actual fact, there's two approaches being taken mostly. There's obviously trying to develop the sort of approach we take. There's approaches where you can try to use genome editing approaches to knock out E6, E7 expression, or one of them, it would be sufficient. And then there's immunotherapies that which have been developed to try and target to get the immune system to recognize E6 and E7.
So,
there's one here.
one here.
Oh, yeah.
A very interesting talk. So my question is, the interactions of these oncoproteins with the virus, do they also decide on disease progression?
Sorry, could you say
Do
that?
they also decide on the disease progression based on the interacting partners that they are interacting at that specific point of time?
Okay, we haven't looked at which, with the exception, with one set of exceptions, there's not really great biochemical data to show what is happening at different stages of disease progression in vivo. Okay, you can do it in vitro experiments, and we know which ones are relevant at different stages of progression. But the in vivo in cervical cancer it's been challenging.
Good information with respect to the kind of association of
you know, you can look at the patterns of expression as you go through cancer, and so, like for instance, the PDZ proteins, they're gone when you get to cancer. In HPV-driven cancer, they're really being degraded very effectively by the viral oncoproteins. We haven't looked at the. We also know that the EGFR signaling is very strong in the early stages of cancer development, but we haven't done we haven't done what I would call a proper biochemical analysis. It's more by correlation rather than actual protein-protein interactions.
This is the last question. We can interact with, we can interact with Lawrence during the tea time.
Yeah, Lawrence, fantastic presentation. It was very interesting to see how much you have developed studying this virus. But looking at the good part of the virus and the transforming ability, especially for the X6 and E7, is a little bit like a dream. I think we can subpower the effect of these two proteins, and we can have like a stem cell induction of some cells. So we can use as drivers for stem cell production.
I mean, the characteristics that these guys give is stem cell. They are stems. Many of the markers that you see in cervical cancer that are being driven by E6 have stem cell characteristics. So that we know. But to use them to, I wouldn't want, you'd have a lot of ethical issues in terms of these two guys. You know, a lot of people have been trying to use them as, trying to use the proteins to promote, I just mentioned the immune response. The delivery for doing that is really fraught because you don't want them also to be inducing cancer. So you have to try and make mutations in them that take away some of these key activities, yet retain their structural features, which is not easy.
Thank you, Professor Banks. So this is the end of the first session. Before we head for tea, I have two small announcements to make. First is that everyone who is registered has been provided 2 coupons for tea and coffee. They are requested to use it, one for for the morning tea and one for the evening tea. And the second announcement is that for the participants especially, you are requested to fill a feedback form. You must have been provided a link through email. So before you leave ICGEB, it is our kind request that you give your valuable feedback to us. So now we will start the next session at 11:15, and you are all requested to head for tea, which is next to the canteen. You can follow the arrow. Thank you.